This presentation is going to nominate a person for Nobel Prize in Physiology or Medicine. Our nominated scientist has a new discovery in the therapy of breast cancer.
Breast cancer is the most common malignancy among women and is the second leading cause of cancer deaths in United States. Less than 30% of women with breast cancer can live for more than 5 years after diagnosis.
One of the drugs used to kill breast cancer cells is liposomal doxorubicin, LD. It is an amphicycline antibiotic which can intercalate DNA. It is a cytotoxic drug used in chemotherapy. However, its poor response rates, high frequency of developing drug resistance, lack of selective toxicities results in narrow therapeutic index, compromising clinical prognosis. Also, the high interstitial fluid pressure of solid tumors result in poor biodistribution and penetration of drugs. These defects often lead to incomplete tumor response, multiple drug resistance, and ultimately therapeutic failure.
LD also have several side effects, including life-threatening heart damage, hair loss, myelosuppression, nausea, oral mucositis, esophagitis, diarrhea, hand-foot syndrome, localized swelling and redness, hypersensitivity reactions, radiation recall, and liver dysfunction.
Dr. Han Chung-Woo made a breakthrough in alleviating these problems. Dr. Wu and his co-worker identified SP90, a novel peptide capable of binding specifically to the surface of breast cancer cells. Dr. Wu finished PhD of pathology in National Taiwan University in 1993 and became its adjunct associate professor in 2005. From 2011 to now, he has been a research fellow in Institute of Cellular and Organismic Biology and Academic Silica.
In the experiment, they use a method called phage display which can be used to study peptide-protein interaction. Firstly, breast cancer cell lines like BT483 were cultured. Secondly, the phage clones obtained is identified using enzyme-linked immunosorbent assay. Phage particles were added to cancer cell and normal cell in ELISA plates to check how strong they bind by measuring the absorbance. Results of above procedures shows that phage clones with higher BT483 binding activities are PC34, PC65, PC73, and PC90. In these phage clones, PC90 bind most strongly to 3 tumor cells. SP90 is developed from PC90 and being successfully synthesized by them. This graph shows PC90 binds to tumor cells strongly, but not normal cells. And another one shows PC90 is competitively inhibited by SP90. This picture shows PC90 only binds to tumor section.
Then, they tried to conjugate SP90 onto polyethylene glycol, PEG, which is on the surface of liposome, changing NHS-PEG-DSPE into SP90-PEG-DSPE. After that, they prepared SP90 conjugated liposomal nanoparticles, SP90-LD, which are accumulated in the endosomes of cancer cells to a much greater extent than control peptide LD. They test the uptake of SP90-LD by human breast cancer cells. Animal model is used for the study of ligand-targeted therapy. Breast cancer cells are treated with SP90-LD and a control peptide LD. Dark box show that LD is bound to the cell. 90% of cancer cell contains LD if SP90-LD is used. Double number of liposome is transferred into the cell by SP90-LD. SP90-LD can kill cancer cells with 1/5 concentration than using LD alone. It is found that the tumor volume of mice having breast cancer grows least when SP90-LD is used, and the concentration of doxorubicin in tumor tissue is the highest. In the picture, red dots are the doxorubicin delivered.
In short, Dr. Wu and his team identified SP90, a novel peptide capable of binding specifically to the cell surface of breast cancer cells, both in vitro and in vivo. Linking SP90 to LD increased the therapeutic efficacy in mice with human breast cancer. Quantification and visualization of doxorubicin levels also revealed increased drug concentration in tumor tissue targeted by liposome, highlighting the enhancement in both delivery and penetration of doxorubicin into the tumor. The results indicate that the SP90 peptide can enable specific targeting of tumor cells in the treatment of breast cancer, as well as to facilitate diagnosis of this malignancy, bringing hope to many breast cancer patients. Since he did a great contribution to the treatment of breast cancer, we decided to nominate him for the Nobel Prize in Physiology or Medicine. That's all of our presentation. Thank you.